can watch it later. All right, so meiosis, we talked about haploid and diploid cells, what that means in terms of having numbers of chromosomes. So a diploid cell, you have a set of homologous chromosomes, one from your parents on one side, one from your parent on the other side. So a diploid cell has this homologous chromosome with this homologous chromosome. Okay, a haploid cell only has one of the two of those. So a haploid cell, you might have two copies of that chromosome, but you only have this one. You don't have the information that's stored over here. So even if you have two copies of this part of that pair, it's still haploid, which is confusing, I know. The information that's held on this chromosome is different than this one. These are not sister chromatids. Homologous chromosomes, they are pairs of chromosomes that are matched up in length. They have the same length roughly. So you can see each one of these pairs has the same length. They have the same centromere position. So when they do duplicate and make sister chromatids, their centromeres line up. They're positioned in the same way. Some chromosomes, their centromeres will be positioned like that. Some like that. Some like that. So depending on where they're linked together is going to determine if they're homologous or not. So this is not a homologous chromosome to this because their centromeres are not lined up. All right. So homologous chromosomes, same length, same centromere position, and same gene locations. So if we have a pair of homologous chromosomes, you have about the same length, make those more same, about the same length, but they don't necessarily have the same information on them. You can have the gene for hair color here on both of them. They both code for hair color, but exactly what it's coding for can be different. This one can be brown. This one can be blonde. Okay, so they're both coding for hair color. They're both positioned in the same place. They're in the same locus, the same gene location, but they don't have to have the same information. All right, so homologous chromosomes can match up nicely in terms of the type of information and where they keep it, but they have different pieces of information. This is where you get the differences between individuals. You get the mixing of genetic information from your parents because one of these came from your dad, one of these came from your mom. All right, so homologous chromosomes are not the same as sister chromatids. Sister chromatids are when you duplicate these. So if I have, I'm gonna do pink and red just to make it a little clearer which one's which. This is my pair of homologous chromosomes. So in your cells, you have 23 pairs of chromosomes. So you'd have a bunch of these pairs of homologous chromosomes. We can have a short pair with blue and purple, okay? So these are both pairs of homologous chromosomes. When you go through interphase, in the S phase, what happens to your genetic information? It copies, it synthesizes. So now, after we copy our genetic information, we will have a pair of our homologous chromosomes, but they're both in a sister chromatid. Both of these homologous chromosomes made a copy of themselves, all right? In mitosis, one of these is gonna go in one daughter cell, this other one is gonna go in the other daughter cell. This one's gonna go into a daughter cell, this one's gonna go into a daughter cell. So you get a copy of every single homologous chromosome in mitosis. In meiosis, that's not necessarily true. You might just get this, or you might just get this. You get one of the four of these copies, all right? So these are sister chromatids. Which are identical copies, all right? So our homologous chromosomes are not identical in the information that they have, but our sister chromatids are identical copies. Okay, does this distinction make sense? All right, so you have lots of these homologous chromosome pairs. That's all of these pairs in your cells, in your genetic information. Every single one of those in each pair makes a sister chromatid pair when it copies. All right, so now you have your homologous chromosomes. We understand that you only get one of each of these pairs, one of each of these homologous chromosome pairs information when you have an egg and a sperm. Which one you get determines your genetic diversity. Okay, so we saw before that meiosis is divided into meiosis 1 and meiosis 2. We're going through the same PMAT stages that we saw in mitosis. Henry, you taking notes? Okay. We go through the same PMAT stages that we saw in mitosis, but each part of meiosis, meiosis 1 versus meiosis 2, is separating a different thing. So in meiosis 1, we are separating our homologous chromosomes. So in meiosis 1, you either get this one or this one. What did we separate in mitosis? When we copied every single chromosome, we copied every single homologous chromosome, what did we separate in mitosis? So in meiosis 1, we're separating these, right? We get one sister chromatid or one sister chromatid. We're separating this homologous chromosome pair. So you either get this one or this one. You don't get both copies. In mitosis, you get both copies. What did we separate in mitosis after we duplicated our information? What did we pull apart in anaphase? Each part of this pair, right? We separated our sister chromatids. So in mitosis, when we pull things apart, you get this, this, you get this or this, you get this or this, but you get one copy of every single one of your chromosomes. In meiosis, you either get this or you get this. You're separating your homologous chromosomes. They're still paired up in their sister chromatids. And that will make sense in a second. So meiosis 1, we're separating homologous chromosomes. So each cell is going to have either this or this. You don't get a copy of everything. All right, so like I said, it's going to be the same PMAT stages. We're going to forget about pro-metaphase. We're just going to do prophase, metaphase, anaphase, telophase. So starting out, just like in mitosis, most of a cell's life is going to be spent in interphase. That is true whether you are a body cell or a germ cell, whether you're a sex cell or a body cell. You're going to spend most of your life in interphase. So what's happening in interphase? So part of it is preparing for mitosis. It has to make a copy of its DNA. It has to grow. What else is the cell doing in this time?
Is it just like doing what G1, S1, and G2? So it's like synthesizing DNA, growing cytoplasm, and building the cell membrane.
So it's doing all those things, but in the meantime, what are cells for? What's their purpose?
For duplicate or to.
Remember, duplication is just a small part of the cell's life. Your parents don't only exist to make you.
Figure out.
They also have just functions on their own, right? So your parents go to work. They function within society. Your cells go to work too. They function within the society that is your body. They're in tissues. They're in organs. They have a job besides just reproducing. So in interphase, it's doing that job also. That is true regardless of what kind of cell it is. So whether it's a sex cell or a body cell, interphase is where it's going to spend most of its life. All right, so up till now, we've got the same thing as mitosis. In interphase, we grow, we synthesize new DNA, and we just be a cell, whatever that means for that type of cell. Okay, so at the end of interphase, we have our 2 copies of every single one of those homologous chromosomes. We duplicated every one of those pairs in our cell. We're ready to start going through our division process. And our first step is going to be prophase. Now, because we have meiosis 1 and meiosis 2, we have a 1 or 2 after each one of these phases, telling you which part of meiosis you're in. So in prophase 1, we are making our chromosomes, just like we did in mitosis. We're condensing that chromatin into chromosomes. We're forming our centrioles. Yeah.
Is this like, is prophase like when I put that underneath interphase or is that?
Now we're in meiosis. So in our body cells, your cell cycle was interphase, and then you had the mitotic phase. Since we don't do mitosis, this is the meiotic phase. So it's a separate thing than interphase. Prophase is the beginning of meiosis. Up till now, interphase is its own thing. It's not part of meiosis. It's just a cell being a cell. All right, in prophase 1, this is our first step. Making chromosomes, just like we did in prophase and mitosis. We are forming our centrioles, putting our spindle fibers together with our centrioles, just like we did in mitosis. And the thing that's different in meiosis is this last part. Homologous chromosomes come together in synapses. What this means is they are coming together and they're going to sort of jam into each other and they can exchange some information. They can switch off. Maybe we don't want this brown piece over here. We can switch. And now this one will have part of the pink one. This one will have part of the red one. So we can recombine our genetic information. We'll get into more detail with that at the end of these notes. But we come together in synapses. Our chromosomes kind of coil up and get tangled. When these things come together, they form these tetrads. Tetra means 4. So 4 chromosomes. We have our 2 sister chromatid pairs coming together, tangling up in a tetrad.
Isn't Tetra a game?
Tetris is a game, yeah. And it's the reason Tetris is called Tetris is because every single one of the blocks that you can move around is made of 4 squares.
Oh. Is Meiosis in?
Yeah.
That's
all of the Tetris blocks. They're all 4 squares. That's why they call it that.
Is Mel's in quarantine?
No, he just wasn't feeling well. Okay. So our tetrad is those 2 pairs of sister chromatids. So you have 4 total chromosomes mixing up, tangling, exchanging information by crossing over. So you cross over one of your chromosomes with the other chromosomes, and then they can switch off some information. Okay, so this is the first difference. In prophase and mitosis, we are just copying and coiling. We're not doing any mixing. In meiosis, we are mixing genetic information. Questions about this so far? In my opinion, the crossing over process is one of the most complicated parts of meiosis. So if you have questions, that's normal. And we'll go into some more detail with it at the end of this.
So like, if I say like to the chromosomes and centrioles form, like, I just say like regular cell, like regular cell?
Same as mitosis, yeah. Same as your somatic cells. So these are still regular cells, it's just that they are a different category. So your germ cells, your reproductive cells, are still a regular cell. They're just they have a different purpose, right? So instead of your body cells making an identical copy, these ones are switching it up so that we have the potential for adaptation, okay? So they're still regular. This is normal for germ cells, for our reproductive cells. This is normal for them. If they did mitosis, that would be kind of strange. Or they can do it, but that's not their main function. All right, so homologous chromosome pairs, we have this one and this one. These are homologous chromosomes, same length, same size, same genes located on same locations, same type of gene. They form sister chromatids when they synthesize new DNA, when they make copies, and then those will come together, tangle up, cross over information in a tetrad. All right? All right, we ready to go to the next step? Yes. Yeah. The next step after prophase in PMAT comes next. Metaphase.
But there is no pro-metaphase.
No, we're skipping pro-metaphase. We're just going straight to metaphase.
Do it.
All right, so after we've done a little bit of a remix with our genes in our tetrad when we came into synapsis and crossed over, now we're in metaphase 1. Okay, so in metaphase, in mitosis, we had all of the sister chromatid pairs for every single homologous chromosome lined up. Okay, so instead of black, let me put these in the colors that I have up here. Okay, so we had our pink one lined up at the center. We had our red one lined up at the center. Our blue one. And our purple one. Okay, so every single pair of sister chromatids was lined up in the middle in metaphase. In meiosis, it doesn't do that because you don't get a copy of every single one of these. You only get half of it. So this was mitosis metaphase. In meiosis metaphase, your homologous chromosomes line up and pair up. All right, so each one of those homologous chromosome sister chromatid pairs is attached to a spindle fiber, which goes to our centriole.
Yeah, those all different colors.
Yes. So we have pink and red. This is our homologous chromosome pair that synthesized, made a copy. They were both sister chromatids, but you don't get a copy of every single one of those homologous chromosomes. You get one or the other. Okay, and this was our other homologous chromosome pair. We have a blue and a purple that are roughly the same size, have the same type of gene information, but you don't get blue and purple, you get one or the other. You get one of those homologous chromosome pairs. You might get this one from your mom, this one from your dad, but you can't get both from both parents. You can only get one or the other, okay? So the difference between mitosis and meiosis for metaphase is in meiosis, you get one or the other. In mitosis, you get a copy of every single one. Because you want all of the same genetic information. You want to make an identical copy of a cell in mitosis. We don't want that in meiosis. We want to switch it up.
So I could say, you say different genetic information.
In meiosis, yeah. Yeah. So in metaphase 1, we line up our pairs of sister chromatids in homologous pairs. How are we feeling so far? Okay. All right.
I think that we should make a GIM kit.
A what?
A GIM kit.
I don't know what a GIM kit is.
It's pretty much, so you know what Kahoot is, right?
Yes.
So it's kind of like, it's pretty much like Kahoot, but instead of like, but instead of, it's like Quizlet Live and Kahoot kind of, but it's like a different concept. It like keeps on asking you questions, but the questions you get right, you get money for it, and so then you can buy upgrades and then get more money for each question you get, and then like other people can like freeze you and stop your money. It's like really fun. I like it.
Well, I can't do that until you understand the information. You can't answer questions right if you don't know what the right answer is. And it's
We
just a
can
random
learn it,
choice.
though.
No, that's not a good way to learn. Maybe review.
After
Sure.
we learn it, can we?
I think you underestimate how much time it makes to make things like that for your teachers. All right. I'm not saying it's not possible, but we'll see. You guys are going to be making something to explain the differences between these in a project. So you're going to make the thing to show that you know it. All right, so we got metaphase. We're lining up our tetrads. We're lining up our pairs of sister chromatids for our homologous chromosomes. One from each pair of homologous chromosomes goes to each new daughter cell, okay? So instead of lining up our sister chromatid chromatids, so you get a copy of everything. You only get one or the other. So when this pulls apart, what's going to be in my top daughter cell? What colors?
Purple. I can't see the colors. Blue and red.
Yeah, blue and red. Down at the bottom, what's the bottom cell going to get?
Purple and pink.
Purple and pink. So I chose similar colors because it's easier to imagine them as homologous chromosomes to me. And these, they do it by size and not by color, and it confuses me. So in these are still homologous chromosomes. These are homologous chromosomes. These are homologous chromosomes. We're lining them up against their pair of sister chromatids in that tetrad. They are homologous chromosomes, okay? So it's 2 copies of this homologous chromosome, 2 copies of this homologous chromosome. Now they're separated. Okay, so we lined them up in the middle. What comes after metaphase?
Anaphase.
Anaphase. What happens in anaphase?
Apart.
That's when they split apart.
They split apart.
Chromatids split apart.
Yeah, so now it's the same. We are separating our pairs. We are pulling at those centromeres from our centrioles, are pulling apart. They are dividing up our homologous pairs. We still have sister chromatids. We still have these pairs, but we don't have both copies of our homologous chromosomes anymore. Now we have split those up. All right. Anaphase still pulling apart. Metaphase still lining up in the middle. It's just a matter of what you're pulling apart, what you're lining up in the middle. All right, we good on anaphase?
Yes.
Yeah. Okay. So anaphase. Anaphase 1 in meiosis. You're pulling apart homologous chromosomes, which are still attached in sister chromatid pairs. Okay. What comes after anaphase? Telophase. We're going to group up telophase and cytokinesis. Telophase, they get to where they're going. We have our sister chromatids that have reached the poles in their pairs still. Some cells will start to reform nuclear envelopes, start to uncoil their chromosomes. Some cells will just go straight on to the second part of meiosis. It just depends. Yes.
Oh, why are they all one if they don't have a second copy?
What do you mean if they are one?
Like it says telophase 1.
Oh, because all of these happen in succession and then meiosis 2 happens. So we're going to do PMAT twice. So we do PMAT 1 and then we do PMAT 2. So there's going to be a prophase 2, metaphase 2, anaphase 2, but it happens in that order still, PMAT and then PMAT again. So in meiosis 1, the goal is to separate our homologous chromosomes. In meiosis 2, we will separate our sister chromatids. We'll go through these same steps again. It's just what are we separating? Okay. So in telophase, we move everything to the poles. Cytokinesis, we divide it. Each nucleus is haploid because you don't have both homologous chromosomes anymore. You only have this one or this one. You might have 2 copies of it, but you don't have all of the information that your original cell had. You only have half of the information. So it is a haploid cell. Agreed.
I'm so hungry.
Me too. All right. Yes. Questions about cytokinesis 1 or telophase 1 and cytokinesis? All right, so at the end of this, we have 2 daughter cells. They are not the same as the original cell. We switched up our DNA in that first crossing over step, and we only have half of it. So we made 2 daughter cells with only half of the information from our original cell. We still have sister chromatid pairs. We still have more information, and then, or we have, never mind, forget that sentence. We have our sister chromatid pairs, but we don't have our homologous chromosome pairs. We only have 1/2 of our homologous chromosomes. Okay? All right, now, like I said, we're gonna go through all this again. In meiosis 2, the point is to separate our sister chromatids.
Just go onto the Google Slides and then copy and paste the notes that you asked.
It's okay, sometimes a little bit of breathing room to think about things is good. Can come up with different questions in the meantime. All right, so meiosis 2. If meiosis 1 was separating our homologous chromosomes, our chromosomes of similar size and genetic information type, same centromere location, now we're separating the copies of those. We're separating our sister chromatids. Why do we need to separate our sister chromatids?
Because that helps the cell split or divide.
Okay, let's talk about, okay, so after telophase, we end up with 2 daughter cells. One has this in its nucleus. One has this in its nucleus. What happens if these 2 things came together and made a baby? Would I have the right number of chromosomes that I started with?
No.
How many would I have?
4.
I would have double the amount of information still. I didn't separate my sister chromatids yet. So in the end, after I join together my gametes, my end result of meiosis, I want to have 1 copy of each homologous chromosome pair. Right now, I have too much DNA. If I put these together, that would be what it would look like after S phase, not what it would look like as an original cell. As an original cell, I'm trying to make, in the end, I'm trying to make something that has every color with just 1 copy. Okay, I'm trying to make sure that I have 1 from each homologous chromosome pair. I don't want double that. I doubled it in S to make my copies, to make my sister chromatids so that I could divide this up and switch it around. But right now I still have too much DNA. If I joined those together, I would have something with too many chromosomes. All right, so now we gotta separate our sister chromatids. So if I joined the products together, it would make an offspring cell with the right number of chromosomes. So what do you think our first step is?
Interphase. No, prophase.
Prophase. We're still in meiosis. We never went back to interphase. We're still in meiosis. So interphase is its own separate thing. Remember that, okay? So we go back to prophase, but now we're in prophase 2. So in prophase 2, this starts right after cytokinesis. This is happening with these products of meiosis 1. If our cell uncoiled their chromosomes in the meantime, they have to
That would not
recoil.
be good.
It would be, some cells do that, but they have to recoil before they can move. If they did that, they'll reform. If they had reformed their nucleus, they'll break it back down again. Not all cells will do that in between step. They'll just keep moving right along without reforming chromosomes, without coiling up or uncoiling their chromosomes. But now we're going right back into it. So if they need this step, they'll do it. Reform your chromosomes, reform your centrioles, take apart your nuclear envelope. We don't need to duplicate our DNA again, right? We want to have half of the genetic number, our chromosome number at the end of this. So we don't need to duplicate our DNA again. If we duplicated it again, we would still have a problem where after that, after we divided it, we'd have double the amount that we needed if we put the cells together. So no more duplication, we're good, we're ready to start again. Okay, what comes after prophase?
Anaphase. Wait, metaphase, I mean.
Metaphase. So metaphase 2. Now it looks like mitosis. Now we're lining them up along that metaphase plate, lining up our sister chromatids. So in metaphase 1, we had our homologous pairs lined up. In metaphase 2, now it looks like mitosis, where we line up the sister chromatids in one line.
How do we draw it?
I was just about to. So in metaphase 2, we have our centrioles still. So each one of these daughter cells from meiosis 1 is going to go through PMAT as well. Okay, so we would have our chromosomes, our sister chromatid pairs, lined up in the middle. Oops, that blue one snuck in there. No. No, that's right. Never mind. Okay. Oh, that's funny. Okay, so we would have our sister chromatid pairs lined up at our metaphase plate in the middle. We're still lining up in the middle, it's just what are we lining up? Metaphase 1, lining up our homologous chromosomes. Metaphase 2, lining up sister chromatids. Okay? If our goal is to separate our sister chromatids, that's what we need to line up to pull apart. If our goal is to separate our homologous chromosomes, that's what we need to line up to pull apart.
It's still like this.
It can be either way. I'll draw it. Let me change it so that it's the same as this one. So before, we had these lined up in homologous chromosome pairs in metaphase 1. Metaphase 2, they're lined up as sister chromatid pairs. Okay. Now, if we line them up, what comes next?
Pelvis. Wait, if you line them up.
We line them up in metaphase. What comes next?
Oh, and metaphase, anaphase.
Anaphase comes next. After we line them up, we pull them apart. Now this again looks just like mitosis. Anaphase 2, our sister chromatids pull apart at the centromere. Okay, so now we have one copy of each sister chromatid pair. If we separate this now, well, we will in a second. I'll not get ahead of myself. All right, so we're pulling apart our sister chromatids in anaphase. Once they reach the ends of the cell, what do we do next? What comes after anaphase? Telophase and cytokinesis, yeah. Telophase 2. After telophase and cytokinesis, we end up with 4 cells. Okay. So do we end up with all of the information that we started with in each one of our cells?
Yes.
Does this one have all of the pieces that we started with?
Like from the very beginning,
Yeah, this is our very, very beginning. We have our homologous chromosome pairs.
it doesn't have to, so like, yes. Like this is like once, it has all the information, but it's not like same exact number.
So it has one of each homologous pair. But remember, our homologous pairs don't have identical information. They have the same category of information, but the type of information that they have, if it's brown hair or blonde hair, they both have hair color information, but the information they're holding can be different. Okay, so if this is my homologous chromosome pair number 1, and this right here is my homologous chromosome pair 2. Okay, originally in my starting cell, I have pink and I have red. I have blue and I have purple. When I end meiosis, does this daughter cell here have every color? No. It has 1 from pair number 1, right? I have my red, and I have 1 from pair number 2. I have my blue. So I have 1 from every homologous chromosome pair that I started with. I don't have both parts of the puzzle. Why do I not have both parts of the puzzle? What am I going to do with these cells at the end of it? What kind of cells do meiosis?
With like genetic and both sex cells, correct?
Sex cells. So what do I do with each one of these cells? Is that its final form? Is that how it's going to go on to live its life forever? What's it going to do? What do eggs and sperm do? They reproduce, they add together. So you have half of it because you only get half from each parent. So I only want 1 of this pair. I only want 1 of this pair because the other one's going to give me the other half of the information when I form a new organism. Does that make sense? What are we confused about right now?
Where does like the other the 2 pairs at the very beginning?
So what happens, this is our original cell, right? This is our OG cell, okay? It goes through interphase when it copies its DNA. So before it can even start this process, your cell actually looks like that. So we made a copy of every single one of those chromosomes. When it's all said and done, your cells have this information. If it's a body cell, that's what it gets, right? It's got 1 from each pair. Before meiosis 1, we still do synthesis. We still copy every single one of those. Okay? Yeah.
And like after interphase 2, does it like split and then there's like 4 copies of it?
Yeah. So in mitosis, we end up with 2 identical cells to the parent. In meiosis, we end up with 4 cells that only have half of the information that the parent has. And that information can be mixed up a little bit from that crossing over that we saw in first prophase. So this is where our genetic mix-up comes from. You only get half from each of your parents. So meiosis ends up with less information than we started with in each cell because we're contributing half to our offspring. What other questions do you have? What's the difference between meiosis 1 and meiosis 2?
The sister chromatin separated and meiosis 2, and then meiosis 1 is like I can't say it.
Homologous.
Homologous chromosomes separated.
So if we look back to our thing here, this is what it's called a karyotype, this set of chromosomes where you have one chromosome, one and its pair. Those are your first pair of chromosomes. That's what your cells look like at any given time. They have one copy of each of those in your body cells. So in interphase, you make a copy of each of those. You'll have sister chromatid 1, sister chromatid 1, sister chromatid 2 and 2 in your homologous chromosomes so that you can separate them and then separate them again. If we just separated without making a copy, we wouldn't get the crossing over step, the mixing up of our genes on our chromosomes.
You said like the homologous chromosomes separate and copy
For one,
Yeah.
so the copying happens in interphase, right? In the synthesizing phase. They did copy, but that happens in interphase. Whether that is mitosis or meiosis, that copying happens. In mitosis, you're still making a copy of every one of these. You just don't put them together like you do in meiosis. Okay, so let's talk about differences and similarities between meiosis and mitosis. Okay, all right, can I erase the stuff on the board?
Can I take a picture of it?
Yes,
What is 8.14?
it's a section in your book.
Do you want to pose for the picture?
Thank you. All right. So we're gonna make a Venn diagram. We have,
Can you make a Venn diagram
I have no idea.
just make a table and then like make a third thing for like both?
That's a good idea. You can have meiosis, both, and then mitosis. Just make columns like that. Cool. Okay. So in our Venn diagram, we have meiosis, mitosis. Okay.
Yeah.
All right, and then in the middle here, what does this represent? Both, things that are true for both of these processes. Okay, so both of them start with diploid parent cells. I don't think I made this diagram big enough. Okay, both of them start with diploid parent cells. What does that mean? What does diploid mean? Two what?
Two cells.
Nope, only one cell.
Two
Two
ploids.
homologous chromosomes. Okay, so a diploid cell for chromosome 1 has a homologous chromosome pair. For chromosome pair 2, it has a homologous pair of chromosomes. For 3, it has a homologous pair of chromosomes. Okay, so it has both parts of the homologous chromosome pair. Are these homologous chromosomes identical in what information they hold?
No.
No, because one of them came from your mom and one of them came from your dad, and they are different people, right? Okay, so a diploid parent cell has a full set of homologous chromosomes. You have 2 homologous chromosomes, okay? They both duplicate these chromosomes. In interphase.
They both duplicate homologous chromosomes?
Yep, every chromosome gets copied in interphase. It's a very lumpy Venn diagram. Okay, so in meiosis, what we end up with is 4 daughter cells that are not identical to the parent. These cells are haploid. So they only got this one, and this one, and this one. Or maybe they got this one, and this one, and this one. But they only have one from each homologous chromosome pair.
The cell like choose which like their color.
It's random. That's where the genetic diversity comes from. So they can line up randomly. Which,
Like, if so like those are like the hair cell like chromosomes or whatever. Like if one was blonde and one was brown and like I don't know, there's like other colors that would be and like if it splits, is like that specific like
gene.
the specific like order of them like in it? Or like the certain like
So you're talking about the law of independent assortment, which I'm going to talk about in just a second. So which set of those homologous chromosomes, if you get this one or this one, is random, completely random. And so that's why if you have like a parent with brown eyes, if you've got a dominant and a recessive, and you have a parent with brown eyes, but they have a dominant and recessive, they can produce kids with brown eyes or blue eyes. Because you might get this one that was held over here on this half. You might get the big one that was held over here on that half. But which half you get is random. All right. So we end up with 4 haploid cells that only got half of that information that are not genetically identical to the parent because of that crossing over, because we can trade off some of our genes. In meiosis, how many cells do you end up with?
4. Oh wait, regular?
Oh, I have these switched. I'm so sorry. I am so sorry.
2 daughters?
2 daughter cells, yes. What do those look like? Are they identical or not to the parent?
Identical.
Identical to parent. Are they haploid or diploid?
Is diploid? What's the difference between haploid and diploid?
Haploid is I only have this one. I don't have this other part of my homologous chromosome pair.
So it's diploid because it's both.
It's diploid. In order to be identical to the parent, if my parent was diploid, I have to be diploid too, right? In mitosis, you stop after that first step. You only separate sister chromatids. You don't have that lining up of homologous chromosomes to divide. You only divide once in mitosis. Okay. The other difference is in meiosis, you have crossing over. That's that process where we trade off information between our homologous chromosomes. Okay. Questions about mitosis versus meiosis? All right. So this is what they look like. Same thing that we just said, just in graphical form. So in mitosis, you have your duplicated.
Can I take a picture of the diagram?
Sure. In mitosis, you have your duplicated chromosomes. In meiosis, you have your duplicated chromosomes, but they have that initial step of crossing over that you don't see in mitosis. In metaphase, in mitosis, we just have our sister chromatids lining up. We don't have our homologous chromosome pairs lining up in those tetrads, those 4 sister chromatids together. Okay. Once those sister chromatids divide in mitosis, you're done. You have identical copies to the originals. In here, you separate your homologous chromosomes that might have had some switching of information, and then you separate them again to your sister chromatids. So not only do you have half of the information, exactly what's being held on each chromosome can switch up a little bit. All right. So those areas where you get this genetic diversity, where you get individuals that look different, everybody in this room looks different from each other. And that is because these sources of genetic variation, places where the genes can get mixed around. Independent assortment is what you were just talking about, Remy. Rennie, not Remy.
Oh, Miss Cody ever literally called me Remy? No, she called me. I think she called me Remy like half the year.
Miss Joanne for like 3 years called me Carlos.
I didn't want to correct her.
It's kind of hilarious, but also heartbreaking.
And then also like like everyone in the class. I wasn't sure if you could even say it. Like, everyone in the class would like say, "Hey, Remy," like in front of her.
I had a fish named Remy, and so I said the name Remy a lot, and sometimes it just slips out.
You had a fish or you have one?
I had when I was in college. Okay, so you can have independent assortment. The way that those homologous chromosomes line up in the center,
Kind of fish.
is a beta fish. The way they line up in the center in metaphase 1, how those pairs line up is random, and they can independently assort themselves. Crossing over is that first part in prophase 1, where we have our chromosomes tangling up, those homologous pairs tangling up, switching some information. And then, for another source of genetic variation, which one of these offspring cells actually makes it to reproduce with the other gamete is random too. Which exact egg meets which exact sperm is random. So not only did you have the randomness that you added in meiosis, putting those cells together is also random. So this is the law of independent assortment. What this is saying is you have this homologous chromosome and this homologous chromosome. You have this homologous chromosome and this homologous chromosome. Which one you get in each cell is random. This could line up this way, where the reds are on one side, the blues are on one side. This homologous pair 1 was lined up blue-red, or it could be blue-red, and then homologous pair 2 was blue-red or red-blue. Okay, so how those pairs line up is random. Question or just scratching? Okay. All right, so when you do that, you separate and end up with different types of cells at the end, different genetic information being held at the end. That is the law of independent assortment. How they assort themselves, how they arrange themselves at metaphase 1 is random. Okay? All right, this is the crossing over that I was talking about. This is what it actually looks like if you were to look in a microscope, a really fancy microscope. You have these 2 pairs of sister chromatids that are literally crossing over each other. This tetrad joins together, This tetrad joins together at specific points, and they can switch the information. Those points where they join are called chiasma. All right, so say you have these 2 parents. You have these homologous chromosomes, these homologous chromosomes. And depending on what combination you get, you can look like a black mouse or a white mouse. All right, not only do I have the color of fur on this gene, on this chromosome, I also have eye color on here too. When I do crossing over, I cut pieces of that DNA and I swap them. So I can have my black color with my brown eyes, my white color with my red eyes. If I cut these pieces and swap them, in the end, I will have a mix of what genes are matched with what genes, what traits are matched with what traits, okay? So you're just chopping off pieces and putting them back where they belong on the other one. How many of you guys have eaten like gummy worms or gummy bears and you like take the head off and move it to the other one and you like swap heads? Never done that? You can do that. You can bite off the head of a gummy bear. It's sticky on the end. If you take 2 colors of gummy bears, you can like swap their heads around. Yeah, that's essentially crossing over. Well, that's good too.
Like with
Just
Haribo's?
because you're ginger.
Yeah, the sugar-free ones will cause you some serious problems.
No, with Haribo's, I get a stomachache after eating like 4.
Oh,
Yeah.
well then don't eat them. But you understand what I'm saying though, right? The concept is
That's the
there.
thing, it's like,
All right,
so good.
we gotta get through this. Okay, so you can do that with your genes as well, just like people can do that with gummy bear heads. You can swap them. That's essentially crossing over. You're taking equal parts and you're switching them and changing the information that they're holding. That is genetic recombination. Crossing over results in recombination. You're combining them in different ways. Okay. So crossing over, you have this sister chromatid pair, this sister chromatid pair, and crossing over, you have them all tangled up, okay? So you have, literally, they cross over each other. That's why they call it crossing over. This is the tetrad in synapsis. And then after they cut and glue back together, you end up with genetic recombination. Where you now have that. You switched some pieces. Okay? All right. So that's your crossing over, your genetic recombination in very simplified form. That is how you get such genetic diversity with all of these different parts and pieces that have these different genes mixing and changing over time.
My siblings don't look like me at all. That's like one adopted, but I don't.
I don't think you're adopted.
My sister's really tall. She's like 5'7".
But sometimes meiosis goes wrong, okay? So sometimes meiosis goes wrong, and you can have things where you get too many chromosomes. That can happen by what's called non-disjunction. If you're pulling things apart and they don't actually separate and you end up with too many chromosomes in one of your offspring cells, your daughter cells, you can end up with trisomies where you have 3 copies of a homologous chromosome set instead of 2. Instead of a pair, you have a trisomy, you have 3. So that can happen in non-disjunction. Things are not separating. Yeah.
Wait, does that like make you messed up in the brain a little bit?
Yeah. Yeah. It changes the way that your cells can function in systems.
Okay.
So yes. All right. So non-disjunction can happen in meiosis 1. If it happens in meiosis 1, you end up with 2 copies of your daughter cells that have 3 cells instead of half of it. In meiosis 2, you can also have non-disjunction. If your sister chromatids don't separate, then you end up with 1 with 3, 1 with 1, and 2 with 2. So depending on when it doesn't separate, you can end up with different combinations of numbers of chromosomes. All right. But the important thing is non-disjunction is when chromosomes don't separate, whether those are pairs of homologous chromosomes or sister chromatids. Non-disjunction, they're not separating the way they should. All right, we take these cells and we do fertilization. We put them together. We end up with our little baby, our diploid zygote. That is the end of your notes.
Yay. Oh wait, no.
It was pretty long, I know. We didn't get as much done on Fridays or on Tuesdays as I wanted to. Okay, so what we're going to do now is I'm going to give you a couple of things. I'm going to give you a gizmo that's going to be for homework to practice all of this because I know that it's a lot of steps. I cut off about half of the gizmo. It still seems long, but I took out a lot of it for you, I promise. Due Tuesday, yes. I'm also going to give you a project description. You are going to be next week doing a project in which you compare and contrast meiosis and mitosis in a creative way. Yes. Tuesday, well.
Any group project?
Oh, for the gizmo, Tuesday before class. So you can turn it in paper or you can turn it on the website. We're gonna go over it in class together though. Hogan, what was your question?
Wait, do we have real school?
Is the project gonna be like for just one person?
We're gonna do a partnered thing and I'm gonna do random partners. So it will be with a partner. You have options for what you can do. You can write a newspaper article, you can write a poem or a song, you can create a model, like a physical 3D model, you can write a storybook, you can make a political cartoon, or you can make a stop motion animated video. So depending on what you and your partner choose, you can do different things. But there are guidelines for how you would do those things, which are on your project description. That is linked on the Week at a Glance. It's also going to be, it's not yet on Blackspot. But for now, while I do the random partner assignments, go ahead and get started on your gizmo. Also, I wanted to say that I'm sorry that the quiz was not on Blackboard for Tuesday. It was supposed to be, but Blackboard changed their settings so that assessments automatically don't publish. Which I was on. I'm sorry about that.
I tried to speak, and I don't know what I'm supposed to write.
Oh, what's the gizmo?
I feel like I started my draft and I.
Maybe not your satanic ritual.
If you just
because
search
you're
gizmo in your email, it's just
Anything besides ginger joke, I don't know.
Your satanic rituals, because you have no
Did you
soul.
say Saipan?
Sorry.
I mean, say it's the same.
Satan.
It's the same.
all right. I'm gonna be right back. I forgot my bag of numbers.
At first I thought it did.
I didn't say Titanic. I said.
I know, at first I thought.
Johnny, what boat should I put on
What
buying
boat?
I'm buying like a 90 picture of the top portion of the boat.
That's
We should
better.
put
Yeah,
that-
Johnny, what boat should we use?
You don't have any good people that want
I know, that's why I said what boat should I get.
You also buy one,
Spoiler.
like what?
No, I don't want to wait like a flat boat or something like that.
Don't Bladesmen like make other
I don't have any emails from Gizmo.
either one of those. Where did you get your
On Microsoft.
I don't have any emails from.
15 minutes. until class.
Hey, John, when are you taking your boat over to our house?
All right, so on Tuesday, what we're going to be doing is we're going to go over the gizmo together, and then you'll have the rest of the class period to work on your project. On Friday, you'll have about half of it to work on your project, then you're presenting your project. So you're going to polish it and finish it off on Friday. You're not doing your whole project on Friday.
We know our partners now.
I will tell you in just a
Will
sec.
Friday be the last day of school though?
Yes.
So we'll have to turn it on like Saturday or Sunday?
No, because you are presenting it at the end of class on Friday.
Yeah.
Oh, okay.
Jacksonville.
And then I can't, where's the Gizmo? Do I just look up Gizmo and try to find it?
Yeah.
I don't have
If you go to your week at a glance, or if you go on the Blackboard, there's an assignment for it.
Alright.
Where did you get it?
Whoa, why is my computer saying it's going to restart?
Last day.
Okay.
Last
No.
All right, first partners, we have
Oh, you mean
Hogan and
Section 7.
Rennie. You're with Hogan, Rennie, and Hogan. You guys are partners.
What was it for your birthday?
No, it was just out of nowhere. My dad just comes home with a boat. And I was like,
All right, next is going to be Gabe and Carlisle. I'm switching it up so you guys get new partners. Learn.
I've never been with Facebook.
Well, we'll find out who you get. Because you're up next, Henry and Avery.
Freak.
offer.
I have to. I don't want to argue with somebody online. That's gonna suck.
All right, Eliza.
Wait, my partner is
I know, but we're starting working on it on Tuesday, so we're okay. All right, Eliza, you are with Mills.
Johnny,
I never feel like
Trevor, you are with Johnny.
It's gonna be Faisal.
William, you are
Hazel.
with
Calling it.
Basil.
Boom. Call it.
That was the only person left.
Okay.
Everybody has a partner, right?
Does Gabe have a partner?
Yes, Gabe has a partner.
I don't have a partner.
Yes, you do. You have a Trevor.
no, I thought he was with me.
No, you are with
I'm actually
Avery.
with Henry.
You are Henry. You are with Avery.
Maybe me and Trevor could switch.
Nope,
Oh,
that's not how assignments work.
that'd be really fun. Be cool.
Henry, do you have any reference for a boat that I should right now? Do you know?
All right, I'm gonna hand you your project.
90s?
Put it on greeny.
Actually, do you want your project description now, or do you want me to give it to you next week so you don't lose it?
Greeny.
Next week.
What's greeny say?
Next week, school next week.
What greeny say?
What are you saying, Hogan?
I'm probably gonna be at school next.
You're probably gonna be what?
Completely under.
I can't hear anything you're saying. Guys, can you be quiet for a second? Henry or Hogan, what are
Probably
you
be at school next week.
saying? Great. If not, we'll do it over Zoom. It'll be fine.
Okay.
But you guys, the project description is still linked on the Week at a Glance, so if you want to take a look at it, it's there.
Depends on your actual job. I already made a stop motion thing for the other project, but then I realized it's on paper.
The flip book?
Not the flip book, the superhero. I made one for the license now.
Gotcha. Well, then you've got plenty of practice.
Miss Garrett, we did the grass boarding on Wednesday.